Thank you. First of all, thank you very much for coming, and thanks to the ISCB committee for giving me the opportunity of being here. The interactions and ligand binding from protein subfamilies to functional specificity. I worked on together with David Valencia, Florencio Pazos, and Alfonso Valencia at the Strategy Biocomputing program at CNIO. So, as you well know, the internal organization of protein families is shaped by the sequence divergence related as a consequence of speciation, gene duplication, and deletion events, and as well by the evolutionary selective pressure exerted on each protein in accordance with the specific function performed. Just as a reminder, multiple sequence alignment provide the opportunity to compare equivalent positions among different proteins and observe which amino acid changes has been allowed or disallowed during evolution. The most used example is fully conserved positions, which pinpoint to important residues for the function and the structure of the protein family. Also, pattern of conservation is those positions that follow a subfamily-dependent conservation pattern. A commonly working accepted hypothesis is fully conserved positions are related with those functional features to all the proteins in the family, whereas these other positions would be related with these features distinguishing with one family from the others. That's why these positions has been termed specificity determining positions. So a variety of computational methods has been developed to detect both protein families and specificity determining positions. I would call them SDPs. And following Rausell et al., these methods can be classified into three major categories. Those methods that use phylogenetic trees as a guide, those methods that uses the correlation between the position similarities with the sequences similarities in the alignment, and those methods that uses multivariate approaches as the well-known sequence space. This last type of methods are very suitable when you want to assess at a time proteins of families and SDPs. Why? Because position weighs the separation between the sequences in the family, and at the same time, the protein similarities with the contribution of the positions to that segregation. In this work, we have developed a new multivariate approach to the families and SDPs, which uses multiple correspondence analysis behind. So I will explain a little bit of the work, won't enter too much into the detail, but the conceptual steps. So starting from a multiple sequence alignment, the G-square distance, which is a proper distance to deal with qualitative data, to decompose the internal variability of the alignment and to obtain at the same time what we call the protein space and the residue space. In these spaces, in the protein space, each protein in the alignment is represented as a vector, or here as a point. And in residue spaces, its residue position is as well represented as a vector. Once we have decomposed the internal variability into these spaces, we assess which amount of information can be considered formative or noise. And we assess that through a Wilcoxon-Rankin test, the optimal number of axes to be retained. So in this space, we assess a clustering of the proteins through a Cummings algorithm and detect the optimal number of groups through the Kalinski-Harabatz and obtain those groups of sequences that are going to be regarded as protein subfamilies. Then, thanks to the MCA property, this link is analytic. The link between the subfamilies and the residues is analytically established. So we detect automatically which are the positions associated with these subfamilies that are going to that are going to be regarded as the specificity determining residue. So, in this work, we want to evaluate the functional constraints that drives protein family evolution by assessing the correspondence between functional features with the organization of protein subfamilies and their related SDPs. Particularly, we're to concentrate on two main aspects of function, protein function. That is the catalytic mechanism in the case of enzymes and the specific binding to small ligands, and importantly, the interaction with other proteins. To assess this correspondence, we're to check how is the correspondence between the protein subfamilies and the different enzyme classes stemming from the enzymatic condition, and how is the correspondence between the subfamilies and differential interactions for yeast and human derived from small-scale experiments. In the case of SDPs, we're going to check the correspondence with these functional features in a structural way, that is, with protein structure, and see how they relate with important residues stemming from the catalytic cytolase and the structural binding to small molecules, and in the case of protein interaction, with assessing the association to protein interfaces. I won't enter into the details, but we were extremely careful in deriving these data to have extremely reliable functional information. So let's see how the method works. These are the results for the family of class 3 aminotransferases. We start from the alignment and obtain the protein subfamilies. You see here how these subfamilies relate to differential enzyme classes, that is, to differential substrate specificities, and then the corresponding SDPs are mapped in a structural representative of the whole family, in this case the human ornithine aminotransferase, bound to pyridoxine. This is a homodimer, which is shown here in gray sulfur and brown cartoons. And the ligand is represented in red, and the predictions are colored in green in that subunit, and in yellow and violet in the other subunit. If you, not a unique perspective to see all that, so we show a small movie, but basically the predictions cluster around the ligand binding site, and importantly in the homodimerization interface. Actually, Markov et al. Have mutated of our predictions and show that these positions were the main determinants of the specificities. It is curious that one of these positions, despite in contact with the ligand, is in contact with another of our predictions in the other subunit, pinpointing to a possible role of the homodimeric interface in tuning the specificity for the site. Let's see a small movie. So here you can see how they cluster in the ligand and the small predictions in the subunit, how they are clustered. So, subfamilies and SDPs also relate to specific interaction interfaces. In this case, we show the ETF-TDP family of transcription factors, and this family is characterized because the E2F factor has the ability of homodimerize or heterodimerize with TDPs here in yellow, leading to differential DNA binding affinities. Here you have the list of the different interactions for this family. So when we get to this family, we actually obtain both families in an answer, that of the ETF and TDP, and we map this SDP representative structure of the family, in this case the head of the human ETF4 in yellow and the TDP in blue. The SDPs in red are not only in but mainly in the interface, in the heterodimeric in a way. These examples are fine, but we want to address these problems in a large scale model. So for this methodology to large scale collection of PFAM families and 1,000 eukaryotic proteins. So let's see the results obtained for assessing the correlation between protein subfamilies with enzymatic classes and differential. We obtain information, functional information in this case, for 150 protein families in the case of the enzymatic classes and 72 protein families of the interactors. So in this case, so the figure represents a rock space. That is the sensitivity and the specificity, and we show the results in a rock space in a bidimensional histogram, in such a way that in each bin it represents the percentage of the family that play this pair of values, right? So, for example, here, 9% of the 150 proteins families we tested. So it can be seen. Okay, let me explain a bit more this. So with a sensitivity of 1 are the families for which all proteins with the same functional level belong to the same family, and the one in the specificity of 0 are the families for which all the proteins within the same subfamily have the same functional level. Can be seen there is a very good agreement for both data sets, and importantly is to see how the figures, the data set of differential interactors are integrated in a coherent manner in the organization of protein subfamilies. It's true that at a level similar of what might be expected in comparison with the, because the biochemical information is much higher. So once we have checked this relationship regarding the protein families, we concentrated on the specificity determinations and their structural correspondence with important functional regions. For that, we derive a non-redundant set of families, a structurally non-redundant set, according to a scope, and obtain a number of families which it was possible to recover that structural information. First, we check the structural distance in terms of these SDPs to the ligand binding site and to the interface. That is, these are just for those proteins that display both kind of functional regions. Establishing a contact distance of 8 angstrom, we can define 4 regions. Positions that are close to both functional regions, positions that are close to the interface but far away from the ligand binding site, and conversely, and also some SDPs that are not close to any of those regions. That may be because they are full positives, or that's because we lack of the structural knowledge, which is incomplete, as you know. It is also represented that percentages for the concept positions, which in general are much closer than the SDPs, but also with a similar distribution. So, a more stringent experiment is not to determine distance, but which predictions are actually functional residues. That is, that are in the binding site of the ligand or in the protein-protein interface. So in this figure, we represent the percentage of SDPs which are in functional regions compared with that percentage for the whole protein, that is the background. And each bar are the results for each of the protein families we studied. So assessing a Wilcoxon reaction test, we saw that the enrichment in functional sites for the SDPs was significant, and more significantly when we restricted to the surface, right? So later on, we wanted to split those, just distinguishing between those positions that fall in the ligand binding site and those positions that fall in the interface, which the site is represented in blue and the interface still in black. Okay, and in green is the overlap, which is observed in the background, not too much, and in some cases for the predictions. Again, both enrichments were shown to be significant. But we still wanted to distinguish which properties of that interface were acting as a signal, as an evolutionary signal for the SDPs to be injected. So we distinguish different cases according to different functional regions. Proteins that display only a ligand binding site, proteins that display only homodimeric and heterodimeric interactions, and a combination of them, and assess again the enrichments. So what we saw is that the main signal from that enrichment in interfaces stems from the hetero interactions. That is, SDPs are especially related with this kind of regions. We didn't find a significant enrichment for the homo and intra interface, although we cannot disregard this association at the light of the results observed for the different cases. So, next, I wanted to assess how these results are dependent of the method used to derive protein subfamilies and SDPs. For that, we choose a number of methods that also assess SDPs, representatives of the three major categories I showed in the beginning of my talk. That is another method that uses a deliberate approach, like sequence space. Another from the method, mutational behavior method that uses the positional correlation with global family, the evolutionary trace, and the combinatorial entropy method. Those methods are not fully comparable, right? And our purpose here is not to compare their performance, because they actually are not comparable. What we want is to reproduce our analysis with these different results and to see if they lead to the same conclusions, right? So, assessing again the different cell enrichment for the different types of functional regions, we observe the enrichment of SDPs in ligand binding sites and the interface as a whole is very robust. The association with the hetero interface is again reproducible with other methods, and the association with the homo and intra interface remains still inconclusive. So the conclusions of this work are that we demonstrate the crucial role of protein interactions in protein evolution driven by functional specificity. And it extends the conceptual framework of SDPs to a more comprehensive definition of protein function. The observation of the individual cases allows to propose that binding specificity evolves by selecting key residues differentially conserved in the subfamilies as pivotal points indicative of binding with their effectors. However, a number of important questions are still open, like the association of SDPs with homo and intra interfaces, but importantly with permanent and transient interactions, and other kind of situations. Recently, Fred Davis and Andrew Jolly published a work in PLOS Computational Biology where they concentrated on those cases that displace at a time a ligand binding site, and an interface. And they concentrated on the overlap in these two functional regions. Characterization the positions that map in this overlap, they reached to the conclusion that they are not spatially conserved, which in principle could be controversial with our conclusions. Notwithstanding, they studied consistency, and it could be harmonized with our results, considering that SDPs, even if they are differentially conserved amongst subfamilies, are not fully conserved. Okay? Just to show that there is a number of important questions still open. So more work and more data is needed. I want to thank especially Alfonso Valencia, the Pathos, with whom I've worked nearly 5 years. I'm pretty much happy for this. The one who has taught me to think in evolution. Then I want to thank for his help in retrieving those functions associated with ligand binding. I recommend you to see the 5V database that he has developed. It's just great. So a poster here. You can talk with him there. And Olivier Listar from the Baylor College of Medicine, Jackson Tippin and Chris Sander from the Memorial Sloan Kettering Center, with the results and the software to use the methods and apply it in this analysis. Members of the CNIO Structural Biology and Biocomputing Program for discussions and help. Here are the fundings. Also thanks to the National Science Foundation at the ISCB for the fellowship. Thank you for your attention.
Okay, we got that first one.
So I didn't quite follow discussing other work. Do you expect these SDPs to be under unusual selection pressure, the possible purifying?
Excuse me, I didn't understand the question.
Do you expect unusual evolutionary selection on the SDPs? That's kind of, I think that's related to what you were saying at the very end.
I mean, it's unusual. Actually, it's pretty much intuitive. It's just a secondary level of specificity, but it fits pretty well with the phylogenetic divergence you observe. It's just.
So you think they're evolving neutrally?
Excuse me?
So you think they're evolving neutrally?
Yeah, for sure. So, No, actually what we demonstrate, they are not, because they are conserved differentially. So actually there is an evolutionary pressure for them to be conserved. And this is why we find a functional association. If they were neutral, there would not be a conservative signal, and it would not make sense their functional association.
But that's within a clade, not between clades, though, is that right?
Excuse me, I didn't hear you.
That's within a clade and not between clades?
No.
It's okay.
Okay.
Is there another question? If not, then thank you very much again.